Okay guys, I'm going to try making a Camtasia video for your structural standoff, as you can see here. And what I'm going to do is I've got the levels of protein structure labeled over here on the left hand side, and you'll notice that how it seems like it has a 1 degree, 2 degree, but that's not a degree sign. It's a shorthand for primary structure, secondary structure, tertiary structure, and quaternary structure. And so what I'm going to do is I'll eventually put a depiction of the level here, and I will discuss the bonds and the interactions on this side here. Simple thing to know when you're talking about levels of protein structure is that at each level, from 1, 2, 3, and 4, as they increase, they also increase in the number of bonds and interactions by that same number. So when I talk about the primary level, you should expect to learn one type of bond or interaction. The secondary structure, there will be 2 types of bonds or interactions. Tertiary, 3 types of bonds and interactions, and then quaternary, something to do with 4. So fairly simple, fairly straightforward. And what we'll do next is we're going to talk about the primary structure. And primary structure is extremely simple. It is simply a sequence of amino acids connected via condensation reactions by a bond known as a peptide bond. Okay, and so I'm intentionally sort of offsetting these a bit, but I'm trying to build this primary structure protein. And to sort of indicate to you where the peptide bonds are, I'll draw in the little lines. So, well, once the pinwheel goes away, and you'll understand that these are the peptide bonds happening in sort of a crude sense. You get the idea that this is a peptide bond. So when it comes to primary structure, simple way to remember is that primary structure has one type of bond. So we've got primary, and it has the peptide bond. Okay, so again, I'm not teaching everything to y'all because you guys are going to do this on your own, hopefully. So primary structure is the peptide bond, so whoever is dealing with the peptide bond, you understand what's going on here. And so what I'm going to do is I'm going to sort of group these up for you, and I'm going to put them on the other page so you can sort of build what I'm talking about and you can understand what's going on. So let's copy. I know that you don't really care that I'm saying this out loud, but I am. All right, so primary structure, okay, and you can clearly see it's sequence of amino acids connected by peptide bonds. Dr. Higgins, can you please put that in there for us? I'm working on it. I'm working on it. So the number of bonds are going to be polypeptide bonds. That's all you need to know. Sequence of amino acids, polypeptide, held together by polypeptide bonds, and hopefully the primary, the group, or the person or the group, the pair that is doing primary structure, I'm hoping that you will include something about amino acid structure. And bond structure, obviously with the polypeptide bond. So amino acid structure and bond structure are going to become important for your discussion. And again, you're going to have to show this in class. So primary structure, polypeptide bonds. Now, I'm going to move on to, we'll just copy and paste this. I'm going to move from here. That's not what I wanted to do. From primary structure to secondary structure. And secondary structure, as we said, you should expect there to be 2. Okay, so spelling is simple. When it lets me do it, it's not letting me do it. Secondary structure, oh Lord. Oh, stop it. Okay, so secondary structure, and within secondary structure, we're going to have two types of bonds. The peptide bonds don't go away. So you're always going to have peptide, but the second type of bond is my favorite type of bond, and that's the hydrogen bond. Okay, so secondary structure is going to be peptide bonds, and they're going to be connected by hydrogen bonds. And for the sake of demonstrations, I'm not going to put in the peptide bonds. I'm going to assume that you understand those. Now, the thing about that primary structure is that hopefully the secondary structure group is going to explain and describe two distinct types of secondary structure as I'm attempting to show you here. This is, I'm representing the beta pleated sheet, beta as in the Greek letter beta, and you can see here that I'm trying to show you here that these individual amino acids are still connected via peptide bonds. However, what isn't as indicated, which I'm about to try to show you, is that in fact they are connected by hydrogen bonds. But I've got to adjust this to sort of satisfy the idea that this bond is temporary. Here we go. And again, individually weak, but collectively strong. And so essentially here, I'm hoping that the group that does this is going to be able to explain to us what is causing the hydrogen bonds to form and how is it that they're holding together our secondary structure. Now, and I'm approximating and guesstimating the positions of these, but the hydrogen bonds are the second, and I'm pointing to the screen, you can't see that, and I probably look funny. That's one version. The second version is going to be called the alpha helix, and this should be reminiscent of the discussion of the double helix and how things sort of spiral. But once again, the indication here is that peptide bonds have not gone away. They are simply, they're assumed here. And if you guys can sort of see, what I'm attempting to do is coil it as if it's coming at you. And I'll try to open the helix up a little bit so you guys can sort of see that. And it's trying to, it sort of causes it to spin and to form a helix. And I think this is kind of a fairly decent demo. Well, I like to think that it is, but this is my first time doing this kind of a demo. And I suppose in a very generalized sense to satisfy the curiosity of what y'all are thinking, you know, you could very easily just sort of make, that would be the alpha helix and this would be the beta pleated sheet concept. Okay, and I'm not going to talk about all the details here. That's what you will do in class. But again, this structure is being held together by the peptide bonds haven't gone anywhere, but they're being held together by the hydrogen bonds. What's important to note here, secondary structure, there are two types of bonds, but there are also two shapes. And I know someone's probably asking me to repeat the shapes, so I'm going to actually type in the names for you as soon as I move this hydrogen bond, which is, there we go, was not cooperating. Okay, so we have the alpha helix. That's not where I want it. Alpha helix, which is this guy to the right, and you can of course abbreviate that, the alpha, as a typical Greek letter alpha, and over here it's beta. I'm not on the smart board, so you can see my issues with attempting to use Greek letters. I'm sitting in my apartment. Alpha helix, and the other one is known as the beta pleated sheet. and I'm sure that you guys are very capable of reading about this, but this is to sort of show you the basics beta pleated sheet. Okay, so there's your secondary structures. So what I'm going to do is I'm going to, once again, group these up, and whoops, I don't really want to stop it. All right. So again, primary structure, peptide bonds. Secondary structure, peptide bonds haven't gone anywhere between the amino acid monomers, but it's the positioning of these monomers that allows for hydrogen bonding to occur. So giving them this secondary level of organization. So we can move these back to our overall table here, and you can see, I'm going to shrink it down, of course, what we're talking about when we deal with the, actually it didn't turn out too bad. It's going to be the primary and the secondary structure, and I know it appears that I'm just droning on and on about this, guys, but this is so I don't have to lecture it in class, and it's to help you when you are doing your presentations, so that this is a ready source as if I had already lectured on it to you. My apologies, I'm jumping ahead to tertiary and quaternary, which I should not have done. Okay, and paste. Okay, so polypeptide bonds. Here the main goal is we have, I'm sure you can fill it in as well, and you can, I'm sure you roll your eyes at me if you'd like to. We've got peptide and hydrogen bonds, and there are two structures, the alpha helix structures, the alpha helix and the beta pleated sheet. Simple, nothing overly complicated about this. So again, trying to summarize the diagrams. Okay, third or tertiary structure clearly is much different, and there's a few things to pay attention to here. What did I do? There we go. Tertiary structure, the third level, so obviously we're going to be dealing with a third type of interaction, which shouldn't have eluded you at this point, but the other significant thing that you want to pay attention to is that at the tertiary structure, this is when a protein can take on a three-dimensional shape and at that point can function as an enzyme. And this is extraordinarily important along the lines of form and function on how this works. So let's see, we've got tertiary structure and the bonds that we're concerned about. Again, the peptide and the hydrogens haven't gone anywhere, but we're adding the third type of interaction, and the third type of interaction is going to be R group interaction. And hopefully we'll understand what the R group means and is when the primary group goes. And I'm sort of harping on this for primary simply because without the amino acid structure, none of this is possible. So and understanding what an R group is a moot point. So primary, peptide, secondary, peptide and H bonds, and third is R group interactions. But we've also said here that at the third, it's fairly important because we get a 3D shape and enzyme functionality. Okay, and that's extraordinarily important. And typically we think of the tertiary structure as a globular structure. So I'm not trying to hold true to what we were showing before as the the peptide bonds and the hydrogen bonds. I'm essentially attempting to just make this globular structure, and this is literally when the alpha helix and the beta pleated sheets sort of fold over on themselves and interact with each other, thus increasing the structure. And I think you can understand what I've just created here is a glob or a globular structure group. And so hopefully when you guys talk about this, whoever has this group, you can talk about the ideas of it having nooks and crannies, and that might seem kind of silly, but it has these little pockets, these little areas, and they're not really little, they're fairly significant areas of, I'm trying to get my arrow happening and it's making fun. Okay, what we want to do here is pay attention to the idea that there's concavity and there is three-dimensional structure associated with this. And so as I fill in, I'm going to draw your attention to where we might see like a little pocket, and that's like right in here. Okay, this could be a little pocket for another molecule to fit in. And so let's see, we can make, all right, we'll take this one, change its color, and we're going to pretend that this is a substrate, or this is a molecule that's going to be acted on, something that needs to be broken down or acted upon by this tertiary enzyme. And we're just, no, what is happening? This is scary. We'll make it purpley bluish or something. Okay, and so this molecule can fit into this little active site, if you guys get the idea. So this molecule can fit into this little active site, as I've indicated, because of the tertiary structure. And also, this molecule is going to be able to hang out or stay in that area because of the R group interactions and the three-dimensional shape. So, and again, we could make a little cavity on any of these different areas within the molecule, but hopefully the tertiary group is going to be able to mention these things. So I feel like this is a good enough indication of what we're talking about here. So tertiary structure, peptide bonds, that's first, hydrogen bond that's second, and then R group interaction for the third level. And we'll put this back up here to sort of a synopsis. And again, this is in your textbook, but I just figured this would be very helpful, and I'm going to also be able to use this for my biology, my freshman classes as well. So, and I'm actually hoping you guys are going to do a really good job on the model/demonstration aspect, because I tend to use those to teach tertiary structure or teach the levels of protein structure. Okay, so here it's globular protein. Okay, so we will do this. We know that it's peptide, hydrogen. So we've got peptide bonds, hydrogen bonds, R groups interacting, and there's a variety of interactions that you hopefully they're going to talk about in that group. So peptide, hydrogen bonds, R groups. It's a three-dimensional structure and functions, and I use the math functions as an enzyme. So that functions as an enzyme. So extraordinarily important in terms of that. Okay. And now finally we go all the way out to the quaternary structure protein. And I hope that this is helpful. If at any cost it's just me playing around. Quaternary structure is fairly simple and fairly straightforward. Essentially what you need to understand is that it is the fourth level. Most proteins, most types of interactions, most things that happen in our bodies and in our cells are dealing with quaternary proteins, which are multiple tertiary subunits interacting together. So if I group these guys up, and this is one subunit. This is essentially one tertiary subunit, one domain of our tertiary structure molecule. But when we have multiples of these now, I'm not sure I'm going to be able to do the properties the right way. Let's see if I can. Oh, magic. When we have multiple tertiary subunits interacting together, we get what is known as quaternary structure. And quaternary structure is the tertiaries interacting with each other They're capable of complex enzymatic reactions. What I'm attempting to build for you here is a tetramer. Tetra meaning 4, mer obviously meaning different units or parts. And if that wasn't obvious to you, then lo siento, I will never take the idea that you would understand that for granted again. Let's make it that. Command D. And I'm attempting to make the tetramer that we all know as hemoglobin, which is extraordinarily important for most of the functions in our bodies. Any color preferences? There we go. All right, so this would be considered quaternary structure. Okay, and we'll group it. And essentially, quaternary structure, my apologies for not copying and pasting everything else. Quaternary, it's not quaternary, it's quaternary. I didn't name them, so it doesn't really matter if you don't like that or not. Command V. And quaternary, 4th level. And again, it doesn't have to be 4 subunits, it could be 2, it could be 3, it could be 11. Quaternary structure, and that's when tertiary subunits interact. So within this quaternary structure, we've still got the peptide bonds, we still have the hydrogen bonds, and we still have the R group interactions, but now all of those tertiary three-dimensional globular structures are interacting together to form the quaternary structure. So hopefully this is making a lot of sense, and when I fill in the table you can see, or hopefully you can understand what it is I was trying to impress upon you here. And I think that you guys get the gist of all this. And again, there's not much to type down here just because it's, well, I'll do it because that's what you want. There's still peptide bonds, there's still hydrogen bonds, there's R groups interacting, but now it's tertiary subunits interacting to make quaternary. So again, guys, I hope that this has helped. I really anticipate seeing some awesome projects here. Let me go to full screen one more time so you can sort of get the idea, and I will summarize everything for you. So once again, we've got the primary structure, which you can see is the peptide bonds holding together the amino acids. We've got the secondary structure, which are the peptide bonds that have caused hydrogen bonds to occur between the subunits. Thus, we get alpha helices here, or beta pleated sheets over here. When the alpha helices and the beta pleated sheets fold over on themselves, it forces the R groups to start interacting in the tertiary structure, and that tertiary structure protein is also globular, it's three-dimensional, and it gains its functionality as an enzyme. And then of course, if multiple, and let me point out, it doesn't mean that it has to be an enzyme at a tertiary structure protein, but that's when it could start to function as an enzyme. And then, of course, multiple tertiary subunits interacting together is what gives us the quaternary structures as shown here. So this is available to you on the topic. So I hope that this has helped, and I look forward to your structural standoff presentations.